Antibiotic resistance is a global health crisis characterized by bacteria evolving to withstand the effects of antibiotics, rendering these drugs less effective or even useless. Several factors contribute to this phenomenon: causes, overuse and misuse. Excessive use of antibiotics, often without proper prescription or completing the full course, allows bacteria to adapt and develop resistance. Inappropriate prescribing. Doctors sometimes prescribe antibiotics when not necessary, contributing to the development of resistant strains. Agricultural practices. Widespread use of antibiotics in livestock for growth promotion or disease prevention can lead to the emergence of resistant bacteria. Poor infection control. Inadequate hygiene in healthcare settings facilitates the spread of resistant strains. Globalization. The ease of international travel and trade enables the rapid spread of resistant bacteria across borders. Prevention. Antibiotic stewardship. Responsible use of antibiotics, guided by healthcare professionals, helps preserve their effectiveness. Public awareness. Education campaigns can inform the public about the proper use of antibiotics and the consequences of misuse. Improved diagnostics. Rapid and accurate diagnostic tools can help identify specific infections, reducing unnecessary antibiotic prescriptions. Vaccination programs. Preventing infections through vaccination can reduce the need for antibiotics. Research and development. Investing in the discovery of new antibiotics and alternative therapies is crucial to stay ahead of resistant strains. Global cooperation. Collaborative efforts at national and international levels are essential to combat antibiotic resistance effectively. Mechanisms of antibiotic resistance. Mutation in target genes. Bacteria can undergo genetic mutations that alter the structure of target proteins, making them less susceptible to the action of antibiotics. Enzymatic inactivation. Some bacteria produce enzymes that modify or destroy antibiotics. For example, beta-lactamase enzymes can break down beta-lactam antibiotics like penicillin. Reduced permeability. Bacteria may develop mechanisms to limit the entry of antibiotics into their cells, reducing the drug's effectiveness. Efflux pumps. Bacterial cells can pump out antibiotics using specialized proteins, preventing the drugs from reaching concentrations sufficient to kill or inhibit bacterial growth. Biofilm formation. Bacteria in biofilms, which are organized communities of cells, can be more resistant to antibiotics due to physical barriers and altered metabolic states. Altered metabolic pathways. Bacteria may modify their metabolic pathways to bypass the actions of antibiotics, allowing them to survive in the presence of these drugs. Plasmid-mediated resistance. Resistance genes can be carried on plasmids, small DNA fragments that can be transferred between bacteria. This facilitates the rapid spread of resistance. Horizontal gene transfer. Bacteria can acquire resistance genes from other bacteria through processes like conjugation, transformation, or transduction, accelerating the development of antibiotic resistance. Understanding these mechanisms is crucial for developing strategies to combat antibiotic resistance and designing new drugs that can circumvent these resistance mechanisms. In conclusion, antibiotic resistance poses a serious threat to public health, and addressing it requires a multifaceted approach involving individuals, healthcare professionals, policymakers, and the scientific community. Responsible antibiotic use, combined with ongoing research and global cooperation, is vital to overcoming this challenge.